Hello. This begins the Unit 2, Module 4, Chapter 8, Part B, Presentation. The big picture topics for this segment include the repair of damage to DNA and mechanisms and selection schemes for creating mutants. Due to the damaging effects that mutations can have on genes, organisms have mechanisms such as DNA repair to prevent or correct mutations. Correction in this case, by the way, means to revert the mutated sequence back to its original state. DNA damage can be caused by a number of different agents. These agents and the specific damage they cause are listed in Table 8.1 in your textbook. Let's begin by looking at the ways the cell corrects mutation before replication occurs. The first correction mechanism is proofreading by DNA polymerase itself. The replication enzyme can proofread and verify the accurate base pairing of a newly synthesized DNA segment. The enzyme will excise an incorrectly hydrogen-bonded nucleobase, which DNA polymerase senses as a bulge in the ladder structure, and it will remove it and insert the correct nucleobase. Mismatch repair is a correction mechanism that repairs erroneous insertion, deletion, and misincorporation of bases that can arise during DNA replication but escape the proofreading by DNA polymerase. Mismatch repair can also repair other forms of DNA damage. The mismatch repair machinery distinguishes the newly synthesized strand from the template or parental strand. A mismatch is then bound by a specific protein. The segment of DNA containing the mismatch is then recognized, degraded, and DNA polymerase synthesizes a new accurate replacement segment. The new segment is ligated to the rest of the helix by DNA ligase. The repair of thymine dimers caused by ultraviolet light can be accomplished through photoreactivation, which acts through photolyases. Photolyases are enzymes that recognize and bind DNA strands containing thymine dimers. Photolyases have a high affinity for these types of lesions and reversibly bind and convert them back to the original individual thymine bases. These enzymes require visible light and are exclusively found in bacteria. Dark repair also uses enzymes to eliminate DNA damage, but does not require light. The second important mechanism for ultraviolet damage repair to DNA is the SOS system. The SOS response is an inducible DNA repair system that allows bacteria to survive sudden increases in DNA damage. It involves several dozen discrete gene products. Mutants of normal genes are used to study structure, function, and regulation of genes. Naturally occurring mutants are rare, so using mutagens to create mutants in a desired gene calls for a method to specifically select them from a population. Direct selection occurs when the bacteria are cultured in conditions where only the mutant will grow. Indirect selection occurs when the bacteria are cultured in conditions where the growth of the mutant is different from the growth of the wild type. The purpose of replica plating is to be able to compare the master plate and any secondary plates, typically to screen for a desired phenotype. Enrichment is an isolation technique that involves culturing cells and adding a bactericidal agent, such as penicillin, that is only effective against dividing bacteria. The prototrophs will begin growing and suicide themselves upon the penicillin, while the auxotrophs will stay dormant because they are lacking a nutrient that they need in order to start growing. Plating what is left over after the penicillinase addition and the suicide of the actively growing bacteria to the cultures to degrade the penicillin onto enriched media should only allow auxotrophs that survived the penicillin suicide selection to grow. A carcinogen is any substance, any radionuclide, or any type of radiation that is an agent directly involved in causing cancer. The potency of many carcinogenic agents can be assessed using the Ames test. The Ames test is a biological assay to measure the mutagenic potential of chemical compounds. A positive Ames test indicates that the chemical is mutagenic and therefore may act as a carcinogen, since cancer is very often linked to mutation. The Ames test uses the bacterium Salmonella typhimurium that carries a mutation in genes involved in histidine synthesis. The Salmonella strain used is an auxotrophic mutant, and it requires histidine to grow. The method tests the capability of a mutagen in creating mutations that result in a reversion back to a wild type or prototropic state, so that now the Salmonella cells can grow on a histidine-free medium. This slide contains questions to reflect upon and use the presented information.